Welcome to our first MaxCyte expert interview. My name is Leslie Eschinger, and I am the Strategic Director of Marketing Applications at MaxCyte. I will be your host for today's interview-style Q&A session with a familiar guest of ours. Our event today is a follow-up event from a webinar we conducted earlier this year in Q2. During that webinar, Matthias Bozza, a postdoctoral researcher and technology manager at DKFZ Heidelberg, described the development of NanoSmart vectors designed to provide stable expression while causing minimal disruptions to T-cell activity. As a result of that webinar event, many of you viewers had some additional questions for Matthias about combining the GMP-compliant MaxCyte flow electroporation system with the CliniMACS Prodigy automated cell processing platform. We think you will find today's session very engaging, and at the conclusion, you will learn how NSmart vectors are used to optimize adoptive cell therapies and really understand the value of coupling these two powerful technologies to create a GMP-compatible large-scale CAR T manufacturing with NSmart vectors. Matthias, are you ready to kick off the session?
Absolutely. Thank you very much for inviting me, Leslie.
Absolutely. Our pleasure to have you back again. And why don't we go ahead and get started?
Sure.
So, can you give us a brief overview of your NSmart vectors and how they're being used to optimize adoptive cell therapies?
Sure. So the NSmart vector is a DNA vector platform that we developed at the German Cancer Research Center in Heidelberg. It is based on an antibiotic-free selection that was developed at Nature Technology Corporation, a company that is based in Nebraska, US. And that combined the advantages of having an antibiotic-free selection system together with the scaffold matrix attachment regions that we have developed in Heidelberg during the last 10 years. So by combining these two technologies, we generated a vector that is a DNA vector that is non-viral, which is mostly based on mammalian DNA. I would say almost entirely based on human DNA, that can be very efficiently produced in bacteria, that can produce in bacteria without the need of post-processing. Once it can be produced as a normal prep, and once that is introduced into human cells, it can be maintained and it can be replicated from the human cells without the need of accessory viral protein. So what we as I mentioned, we developed these in the last decade almost here in Heidelberg, and only recently, in the last couple of years, we decided to leverage this technology for the production of adoptive cell therapy products with a particular focus for CAR T cells production. And as part of the webinar, I was describing how we use these techs to make CAR Ts.
Right. In your publication from Science Advances earlier this year, you mentioned you wanted to determine if NSmart vectors would be suitable for clinical applications in engineering primary T cells. And as a result, you tested the large-scale electroporation of NSmart vectors in human CD3 positive cells using both the MaxCyte ExPERT GT platform as well as another electroporation platform. So that being said, why did you decide to choose using MaxCyte over the alternative EP device?
So the whole idea of using non-viral system is to shorten the time necessary for producing these products. So as everyone is aware of, when we use viral systems, it takes about on average 20-25 days and is a rather complex supply chain. So what we wanted to do was to really shorten these vein-to-vein time using fresh products by using also in-house GMP facility. And so our decision to use the MaxCyte was really dictated by the fact that by having such a compressed protocol, we really were looking for high viability post-electroporation. And the ExPERT device was a large electroporation devices that gave us the highest viability post EP. And when I say post, I'm referring to 24 hours after electroporation.
Perfect. So then my next question, I think you just answered, was the recovery time from the cells. Did that factor into your decision on what technology to use going forward? It sounds like it did.
Yes, yeah, absolutely. Absolutely. So the whole idea is that we don't want to, by having low viability, we don't want to basically to waste a lot of the material coming from the patient. It's very precious material. And some of the patient, even from leukapheresis, we cannot extrapolate a lot of cells. So we really have to care about the cell's viability and fitness after EP. And this was for me the most crucial parameter.
In the manufacturing workflow you have established at DKFZ, combining the GMP-compliant MaxCyte flow electroporation system and the CliniMACS Prodigy automated cell processing system, what final volume have you been able to achieve using this workflow, and how many patients can you treat as a result?
Yeah, so for the DNA manufacturing, we can produce up to thousands of patients. We produce roughly 2.5 grams per liter, which is from an off-the-shelf product. It lasts for roughly thousands of patients. However, we are also tailoring this process for individualized medicines, where we want to produce 1 TCR or more, up to 10 TCRs in a very short time window, and that's something we are also managed to achieve. Regarding the manufacturing, so we can produce with such protocol about we can manufacture in four products per month, considering that every product takes about 7 days. And when we compare these to lentivirally produced products, is significantly better, because the manufacturing protocol with the lentivirus takes about 15 days, which clearly will improve the turnaround of the patient and the number of patients that we can treat with adoptive cell therapy.
Okay, can you comment on MaxCyte's reproducibility when transfecting NSmart vectors into primary cells, and what implications this has for cGMP?
I think that the device is very robust. I think we do see variability, which is donor dependent, which is expected to be like this. However, when we introduce, for example, a CAR, we can consistently hit more than 60% of the cells. And when we introduce a TCR, it is slightly better, and we can hit robustly over 80% of the cells. This goes from 80 to 99.9%. It really depends from the donor.
Okay. In the 2021 webinar you delivered back in June, you discussed the minimal effects of the NSmart vectors that they had on T-cell gene expression. So why is it important for the functionality of your vectors in the development of CAR T-cells?
Well, we compared what are the overall perturbations of cells that were infected with the lentivirus, or the cells that were electroporated with the ExPERT device. And what we found is that the introduction of the DNA by electroporation has a minimal impact on the cell's homeostasis. While when we introduced the gene of interest by lentivirus, we saw a larger number of genes that were either up or down regulated, which actually made us speculating that it's kind of that the cells recognize that are infected by a virus, and they are somewhat fighting the infection. Indeed, we found marker of these fight upregulated. While when we do this by electroporation, we don't see such effect. And and I have to say that when we just look at the no DNA control, when we just deliver the pulse, we couldn't really find any genes that are massively up or down regulated. Actually, I don't think we can find. I think there are less than a dozen genes that really change, which made us concluding that the electroporation has literally no impact on those cells, while the effects that we see is really triggered either by the DNA, which are very minimal, or by the virus that are more significant.
Great. And then similarly, along that same vein, did electroporation of the cells impact the ratio of the CD4 positive and CD8 positive cells in the populations, or cell proliferation?
No, cells that have been elected, that where we deliver the DNA or that we follow up only also with no DNA. So the proliferation was not affected at all, as well as the ratio CD4 CD8 was not affected by the electroporation. So I would say that the electroporation has literally no impact on those cells, nor the homeostasis, nor the phenotype of the cells. I mean, the changes that we will see in the population are induced by the culturing, so the way we decide to culture the cells. But the EP itself is basically harmless.
Okay. Your team demonstrated that electroporation of the NSmart plasmid resulted in a T cell with more naive state relative to lentiviral infections. Do you believe this would impact T cell exhaustion and the efficacy of the CAR T cell therapy?
Yes. So this comes down to what we mentioned before, right? If we deliver by EP and we use basically an invisible vector, the cells do not feel that they have been modified. So they do not feel that something foreign has been introduced. While if we do the same procedure with the lentivirus, the cells start to feel the infection, start to fight the infection. And as a result, if a cell is busy fighting an infection, is not as good concentrate in killing a tumor, right? While if we have a cell that is basically in its naive state that has been modified with a very gentle technology or a platform, then it's probably more efficient in killing the tumor cells. And that's really what we saw in the results in our hands. That cells that have been modified with an NSmart plus electroporation are much more efficient in recognizing and killing the tumor cells compared to cells that were made with a virus.
Okay, great. In your opinion, will MaxCyte's electroporation approach help with therapeutic applications of the CAR T cells in this regard?
Yeah, I think absolutely. I think that the fact that we can, work at scale, really from small to extra large scale, with such efficacy of delivery and such high viability will certainly help the transition into clinical applications.
And did you experience any technical challenges when pairing the Miltenyi CliniMACS Prodigy system with the MaxCyte GT ExPERT electroporation system?
Yeah, I think it really depends from at which scale we want to work at. I think if we at extra large scale, at extra large scale is not a bit difficult. It's not very difficult because the Miltenyi formulate the cells at 100 mils, and 100 mils is also the upper limit to the biggest MaxCyte cassette, so it's the CL2, which will not make any problem. The problem could be, that you may need to adjust the size concentration, but in terms of volumes, there's not much problem. However, when we want to work in smaller scales, when you want to work with a CL1 or with other cuvettes or other PAs, then there is a problem because then the Miltenyi can only formulate at 100 mils, and then you have to centrifuge and adjust your cell concentration, which clearly makes the problem that we have to open the system. So then the system is not closed anymore, and we need to be creative to find a way to make it GMP compatible.
Yeah, and since you're already on that topic, what approach did you take to enhance the pairing of the two systems to act as almost, a semi-closed system in that particular example you just gave?
We concentrate the cells using you can concentrate the cells using concentrators in bags, such as the Rotea provided by Thermo Fisher, and it is a closed system, so you can basically well bags and concentrate. I think going forward, I think that whenever MaxCyte will release bags with smaller capacity, will make things probably easier to couple the two technologies. In our case, we often had to centrifuge and basically open the system. So we are not really entirely closed. So we're working on a solution to keep the system closed.
That process you just described, was that labor intensive to produce the CAR T cells with the two platforms combined?
No. It was not labor intensive. I think every experienced technician or every experienced GMP technician can make it. It's particularly simple actually because the Miltenyi, it's a rather straightforward instrument that does everything. Does the isolation of the T cells from a leukapheresis. That's very straightforward, and as well as the ExPERT is very straightforward when it comes to electroporation. So the only really tricky point is when you have to adjust your cell concentration depending on which scale you want to work at. That's really the only moment where you may have to open the system or have very fine-tuned your condition with 100 mils.
Okay. Are there any other areas for further optimization for your workflow for the CAR T cell production?
I think we really need to determine what are the working range. For example, it's still not very robust, the number of cells, for example, that you can activate with the Miltenyi in a very short period of time. We're talking about 7 days of protocol, which means that we use 3 days of activation. And sometimes it's already variable, the number of cells that you can generate after 3 days of activation. It's not a huge range, but it's still a range. And then from there we need to then optimize, actually define what are the working concentration ranges in the electroporation as well. And clearly, if we want to even shorten the protocol, this has to be done for every step, for every activation day that we decided to trim. So the idea is clearly to have it the shortest possible, if possible with no activation at all, and that clearly requires some fine tuning of the cell numbers, cell volumes, and DNA concentration for the electroporation.
Okay, and in your opinion, did MaxCyte contribute to the decrease in time required to get your CAR T cells into the clinic?
I think so. Generally, absolutely. I think, as we said, by having a device that can really generate cells with high viability and high efficacy, at the end of the day, it's a matter of how many cells you can produce in a given time window. And if the idea is to make these cells in the shortest time windows possible, then a device that allows you to make very viable and a lot of positive cells, it's clearly helping us in shaving time to this protocol.
Great. And just a few additional questions. What makes the NSmart vector unique or novel?
Well, it is the only only DNA or plasmid DNA based vector that is able to auto-replicate when it's in cells. So basically, there's no other technologies based on DNA, on plasmid DNA, that do not exploit viral protein that are able to be replicated from human cells without either killing the cells or integrating into the cell. So this is the only platform that once it's delivered in cells can be replicated and maintained without being integrated.
And have you tried to transfect multiple NSmart vectors in a single electroporation? And is multiplexing possible with your technology?
Yes, that's where we are leading to. I think multiple transfections is one thing. However, by having a non-viral systems, you may want to combine everything in one because the payload size is not a limitation. We're multiplexing, for example, for delivering of several TCRs. However, we're not really multiplexing in the same transfection. We're multiplexing in, several electrophorations. But sure, I mean, delivery of multiple vectors is something we work on, and it's feasible. And it works very well.
Right. Are you using the NSmart vectors in other application areas or with other cell types?
Yes, we are trying to exploit every cell type, so to say. We are using, the natural application was CAR Ts and T cells because that was it's most trendy. Clearly, we are now working on NKs, but we have applied the vector to modify mouse embryonic stem cells or human stem cells. So we are exploiting the possibility of doing regenerative medicine too, and we are going back a little bit to the principle of all gene therapy, in vivo gene delivery and in vivo gene therapy.
And just as a last question, what are your next planned future studies? Do you have plans to move into the clinic?
Yes. So we really hope to get next year into the clinic. We are trying to acquire funding for a novel TCR approach, novel TCR-based approach for a phase 1/2 combined. And being in this situation where we have a very close relation with the National Tumor Center in Heidelberg, we are involved in compassionate use trials too. So I think next year will be really the year where we will apply inpatient for the first time cells made with such platform.
Great. Well, that's all the questions we have that I had for you, Matthias. So thank you very much. And I wanted to just say, again, thank you for all of your efforts in providing our viewers with your detailed answer. I wanted to thank our event sponsors, LabRoots, for all of your assistance organizing this informative session, and wanted to thank our audience participants for your continued engagement and for providing great follow-up questions from our original webinar event. This concludes our session for today, and we look forward to future events. So thank you again very much, Matthias.
Thank you. Thank you.